Alright, welcome to Chapter 14, everything you want to know about genetics and more. So some of these I went over in class, but I wanted to go over the full thing here. So, first of all, transmission of characteristics from parents to offspring. Sounds really big. All it is heredity, saying you get characteristics from your parents. The science that shows how these characteristics are passed on is genetics. So, talked a little bit about this in class. The two ideas how genes are passed. We used to think as a little bit of blending. So if mom's a yellow, dad's a blue, all the kids are going to be green. We have now found that sometimes that's true, but for the most part, that doesn't happen. Problems with that is predicts over time we would all have green traits that you wouldn't be able to skip traits, etc. The other part is particulate. Parents pass on discrete units that help they're separate. So a little bit. So that's a little bit more like each of the different genes. So the father of genetics is Gregor Mendel. He's the one who played with pea plants, and he found and these pea plants were perfect because they had all these dominant characteristics, and he meticulously wrote all the stuff and looked at one trait at a time and spent years getting all this stuff. Had extremely careful records. So he, with these peas, he self-pollinated peas, and he actually snipped off little pieces, painted, pollinated all these peas, was able to find that egg and sperm came from the same plants, and was able to observe offspring produced for different crosses. So you start off with the parents. So he first would cross two different characteristics like purple and white, and that's the parental generation, and found they were all purple because purple is dominant. That's the F1 generation. They're all going to be heterozygous. So if you think of having a big P, and small p, put those together, you're all going to have big P, little p. They're all going to be heterozygous. And that F stands for filial. If you breed those two heterozygotes together, you get that 3 to 1. So you have 3 to 1 ratio. That's your F2 generation. So, patterns are the key, and this kind of shows you what's happening. And this is showing each of the different gametes so here's your phenotypes, what it looks like. Here is your genotype, somewhat. They just don't say all the genotypes here. These are all heterozygous. And you can see that dominant, round seeds, and the one wrinkled seed, the recessive trait. So, the genetic makeup is the genotype, the appearance is the phenotype. And so, the genotype are these letters, phenotype are the colors. What Mendel found. So, he found alternative versions of genes account for variations in inherited traits. So, basically, you always have two alleles, and there's different versions. It would be big T, small t, small t. For each trait, an organism inherits two alleles, one from mom, one from dad. If the two alleles at the locus, so that's just like the location, differ, one determines the organism's appearance, the other has no effect. And the two alleles for a character separate during gamete formation, again egg and sperm, and end up in different gametes, so that those alleles are going to separate. Alternative version of the genes account for variations, and so a trait is that observable characteristic. So each of these are different traits of the pea plant. And so you can't really see down there, but the allele are the choices, so the versions of the gene. So brown eyes, green eyes, blue eyes, red eyes according to this one. So each of those are different allele, tall, short, axial, terminal. Whereas each one of these is a different trait, so seed shape, seed color, etc. So for each trait, an organism inherits two alleles. We know that from meiosis because we get one from mom, one from dad. So organisms with two identical alleles is homozygous, or pure breeding. It's pure. Can't read this last part because I couldn't get the entire screenshot, but it says organisms with two different alleles is called heterozygous or hybrid. If the two alleles at the fungi or locusts differ. One determines the organism's appearance, the other has no effect. So that's just talking dominant versus recessive. So dominant masks one and the recessive is hidden. And the two alleles are separate. This is Mendel's law of segregation. So here you have your two chromosomes and they separate. And then for gametes, each one of those are going to separate as well. So you can see here's your diploid. You have your homologous chromosomes. They separate. And then each of the gametes has one. And so they're all independent of each other. Little meiosis, all crossing over, go into each of their own cells, and they're each of their own little cells. So that was law of segregation. Law of independent assortment is when two or more pairs of alleles or genes segregate independently. So these genes have no effect on these genes. Probability is you guys know this from math, that this particular event will occur. So if you close your eyes and pick a disc, what is the probability you pick the white one? So you could say it's 1/4, you could say it's 25%, or you could say it's a 1 to 3 chance. And we use all of those during genetics. So coin flip, there's two possibilities, head or tail. So chance that they'll land in either 1/2, 50, or 1 to 1. So the alleles segregate randomly just like a coin flip. You can use probability to predict these genetic crosses. Things to remember. Past outcomes don't affect future ones. So, if the last coin flip was a head, there's still a 50-50 chance the next flip will be heads too. So same thing when you're thinking about kids. I have a son. It doesn't mean now my chances are going to happen more that I'm going to have a female next time. I still have 50-50 chance of having a boy or a girl. Probability works best when predicting a large number of events. This is always true. The larger the number, the larger sample size, the more. So the more flips, the closer results will be 50-50. So in humans, if we're having 1 or 2 kids, we may not get, or even if we have 4 kids, we're not going to get exactly what happens in the Punnett squares. But if we're able to breed plants and we could breed hundreds of offspring, you're going to see more of that ratio. Multiplication and addition. So you always multiply the probability of one event by the probability of another. So what's the probability that 2 coins tossed will simultaneously have both ends, both end up on heads? 1/2 times 1/2 gives me a 1/4 chance. Same thing as saying what are my chances of having a second son? My first was 1/2 chance. If I have another kid, 1/2, so 1/4. So this works in genetic crosses as well. What's the probability of having offspring that will be big T if both my parents are heterozygous? So each egg has a 1/2 chance of passing on a T. 1/2, 1/2. So if I multiply those together, I get a 1/4 chance, which you guys can see in the Punnett square as well. Addition. So to figure out the probability of offspring being heterozygous, so a big T can come from mom, small t from dad, or vice versa. So if I get it the first way, I have a 1/4 chance. Second way, a 1/4 chance. Now I add those together to give me a 50%. Yes, you could probably do these ones without doing the probability, but when we start talking about di, tri, quadruple hybrids, you're going to want to use probability. So, how can you tell a genotype? So the tall plant can either be big T, or big T, small t. What you need to do is cross it with the recessive. That is called a test cross. So you're crossing it with a homozygous recessive, because I know what its genotype is. So if they all are still tall, I had a big T plant. If some are tall, some are short, I had a heterozygous plant. So these are the different, so Mendel came out with all these 1800s. We've now found different parts. So we're going to be talking about each one of these. First thing, environment influences phenotype. Craziness. How does this do this? So we have found that if they that the Siamese cats and Himalayan rabbits have dark colored fur based on the temperature. So the alleles that control the pigment production is only able to function at lower temperatures of the extremities. Crazy. So they have it all throughout, but since they don't have as much temperature in these parts, they have darker spots. Crazy. The hydrangeas, depending on the pH of the soil, will change their color. As far as spectrum of dominance, we have complete dominance, incomplete, and codominance. So it's not as simple as that purple and white flower we saw earlier. Sometimes they're a little bit different. Complete dominance, this is that purple and white flower, this time red and white, but breed the parents, they all are heterozygous, and we get our wonderful 3-to-1. So that is dominant versus recessive. However, sometimes we get incomplete. You mix that red and white, and then all the heterozygotes are pink. They're a little bit blended. And then, so if you breed those two together, you'll get a red, a couple pink, a white. Do the Punnett square. It really does work. There's also codominance. So here's that horsey butt we showed in class. And this is called roan. And you might be able to see it a little bit clearer now, but you get red and white. They're both dominant. The biggest thing we talk about with that is our blood types. So A and B, they're both codominant. So we have these glycoproteins that are attached that are recognize different pieces. So these type glycoproteins will tell you what your cell type is. And we use I for blood type. I don't know why. If someone finds that out, some bonus points. So, blood types. A person who has A, we would write it as I with big A, or IA. This would be your heterozygous one, because this I is your recessive gene, which is the O. So if you have type A blood, you have a whole bunch of little A's. They recognize other A's. B, and you would write it like this, or like this. You have a whole bunch of little B's. The O, they're recessive, so they have two little I's. And there's nothing. There's no glycoproteins. The big A, big B, you have both of them, and that's the only way it can be written. That's the only way that would work. It's like this. So here's the different blood types. And a few more things about blood type of why they don't go. So if you don't have anything, A blood, this one sees A bloods as different, the immune system attacks. So right now, these guys can get along, these guys cannot get along with this A blood. For B, same thing, B blood can go to B or AB, but not to those. AB can't go to anyone, it can just go to AB. The O, it can have everyone. O can donate to everyone. So it's the universal donor. However, it can't take any of those. AB can receive from everyone. And so it's called the universal recipient. There's also the RH hearts, and so you can be RH positive, RH negative. And the only time there's problems is, so right now we're good. If mom's positive, she's always good. If mom's negative and the baby's positive, that's where you can get the baby end up dying because they have all these positive ones and mom's going to make antibodies against it, so the second child be making it against. So we are going to stop there. Alright, we'll stop right here. So these are your different choices. We will stop right there.